Okay, we're going to start with the Corynebacterium. Okay, so aerobic. Gram-positive bacilli. First one we're going to talk about is just general information for the Corynebacterium. And what's the most, I guess, influential or descriptive part of Corynebacterium? Has to do with the gram staining. It's going to be like your Chinese letter looking ones. And so whenever I make notes about it, it's kind of hard on the Word document, but I put like little Chinese letters next to like Cranibacterium when I'm writing down the notes, and things like that kind of help. So gram-positive, and it's either going to be a rod or a bacilli. They are non-spore forming. So your Chinese letters are key information for that. And what's the Chinese letters referring to? Does it look like Chinese letters on the SBA plate, or? What are they referring to when it says Chinese letters? That's going to be on your gram stain. And so not the morphology on the plate, but on your gram staining. And then they're also described as clubbed shape. They're generally catalase positive and non-modal. Now specifically Cranibacterium Diphtheriae. What does that cause?
Diphtheria.
Diphtheria. So it's a diphtheroid. Would have never guessed that, huh? They're small pleomorphic rods. What does pleomorphic mean? Do y'all know if something's described as pleomorphic? So if it's pleomorphic, then it can take on different shapes. So it's not always the exact same shape every time. And then it often has clubbed ends. So are the ends going to be thicker or thinner than the rest of the rod or bacilli?
Thicker.
Thicker. Again, Chinese letters, very important. And it's also a palisade, which refers to it as like being in parallel rows compared to just like randomly thrown around. So diphtheria is the infection that I think you're most worried about. And it can either be an upper respiratory infection or cutaneous. And then the way you can remember this virulent process is C, D, E, F. And so if you can't remember which letters they start with, it's Corynebacterium Diphtheriae. So C, D, and then E, F. And those stand for Cranibacterium, Diphtheriae, and then it blocks elongation factor. And so that's how you can remember the disease process. Cranibacterium Diphtheriae blocks the elongation factor, C, D, E, F. And what that leads to is inhibiting protein synthesis in eukaryotic cells. And then you have an ELIC test, and I think we're going to talk a little bit more about it later. But what that does is determines the toxin production in vitro. There's a vaccine. And so what's that going to do to the caseload? Are you going to have more cases or less cases? There's a vaccine for something.
Less cases.
Less cases, yep. So it decreases the cases due to this vaccine. Now they stain. Y'all know how they stain? Do they stain the same all the time for C. diphtheriae? They actually stain irregularly, which gives it a beaded appearance. And then have y'all heard of your Babe's Ernst granules? Did y'all talk about that at all? So Babe's Ernst. Y'all know what they are? Babe's Ernst granules. They are your metachromatic areas that stain more intensely. And what they indicate is they indicate an increased nutrient reserve. So if you're feeding it on an agar plate, what is that going to increase? Or is it going to be more metachromatic or less metachromatic?
More.
More. So your selective media for the Corynebacterium Diphtheriae is going to have that metachromatic areas more intensely than normal, so that way you can kind of differentiate it. It's a facultative anaerobe. So what does that mean? A facultative anaerobe.
If it has air, it'll use it.
If it has air, it'll use it, but it's not, or it can go both ways, but it actually prefers aerobic. But if there's a lack of air, if there's no oxygen, then it can be a facultative anaerobe, which means it can still produce energy without having that oxygen. So, but the optimal growth is going to be aerobic at 37 degrees. These I should move back one probably. There we go. Now, do you all know the selective medium? So Loeffler's medium. Going to be selective. So you know there's differences in selective and differential and different kinds of media. So your Loeffler's medium is going to be selective and increases the Babes-Ernst granules. And so what does that mean? If it increases the Babes-Ernst granules, what will you see visually?
Spots of more intense staining.
Yes, there's metachromatic staining. And then on your sheep blood auger, you're going to have a small zone of beta hemolysis. And then you've got another important media, and that's going to be your CTBA. CTBA. That stands for cysteine tellurite blood auger, or it's also called the Tinsdale. So cysteine tellurite blood auger. Your Tinsdale. They shorten it to Tinsdale. Now, do you all know whether that is selective, differential, anything like that? It's actually going to be selective and differential. And you're going to see a Tinsdale, what they call a Tinsdale halo. Have y'all seen that, like in the book or anything? What a Tinsdale halo looks like? It really just kind of looks like a little halo around the colony. And then, do y'all know what color they're going to be for C. diphtheriae on a CTBA on Tinsdale? What color are they? They're going to be your black or brownish colonies, and this is due to the reduction of tellurite. Find a nice picture. I'll put it up here. So here on the left, what is that on, plated on?
CBA.
Yep, the CTBA. And then here on the right, that's just going to be on your blood auger. And so notice the difference. They're black on the CTBA and white on the blood auger. And then let me get my chart. I bet they are in now. Okay, so here's the colonies on Tinsdale medium or CTBA producing a brown halo. If it's gram-positive, then you're thinking gram-positive rods and dipteroids. And then you can do a urease test. So if it's positive, it's going to be acid from starch or glycogen, which can be either Corynebacterium ulcerans or Corynebacterium Pseudo tuberculosis. And if it's urease negative, that's your Cranibacterium Diphtheria. So it's really important to write down things that are differential. So like your urease negative would be differential from other types of Cranibacterium. And so that's an important one to keep track of. And now if it's going to be gram stain negative on with a halo on Tinsdale medium, if it's gram-positive cocci, it's either going to be staph or strep, or if it's a gram-positive rod, it's going to be another type of Cranibacterium. So I think the most important part of micro is going in and figuring out which ones, like what are your differential aspects of it. Now, next one's going to be, there's a bunch of different types of the Cranibacteria. I just did a few of them that you hear about a little more. First one's going to be C. Jijincum. And this one, like if you do the different like lab CE and stuff like that, you hear about this Jijincum or C. Jijincum more often than others, I feel like. So it's normal skin biota. Infections. Know any infections that it can cause? Or how about the people that are generally more susceptible? So when you have something that can be a pathogen but is normal skin biota, who is usually affected?
Immunocompromised people.
Yeah, so that's going to be your number one, immunocompromised hosts. Invasive procedures can cause infections. Central line catheters. All these classes make you never want to have a catheter. So central line catheters and prosthetic devices. And usually what disease are you associated with prosthetic devices? Starts with an E. It's a type of inflammation.
Endocarditis.
Endocarditis. Yep, that's usually what is associated with any kind of prosthetic devices. It can also cause septicemia, which is what? Where is it located if you're septic, if you have septicemia?
In the blood.
In the blood. Then meningitis, aesthetic point. And then skin complications. So your C. jejicum is lipophilic. What does that mean? Lipophilic. Can you break it down? What's lipo?
Lipids or fats.
Lipid or fats. And philic, does it like it or not like it? Can it permeate that? It's going to love, lipophilic is when it loves fats. They're non-hemolytic. And it's a strict aerobe. Is that the same or different from C. diphtheria?
Different.
Correct. And then I want to make a comment that most of your results, differential results for Cranibacterium, specifically for the Chichium, is negative or variable. You can't remember every single one of them, but we can look at the what page the chart is on. Should have written it down. Almost there. I know there's one specifically for Cranibacterium somewhere. Page 349 in y'all's book. So really the only thing it's truly positive for out of all those characteristics is lipophilic. But nitrate, urease, fermentation, acid production from glucose, maltose, sucrose are all negative or variable. So that's what I mean by most of the results are negative or variable. And then for your antimicrobials, what are antimicrobials?
Like antibiotics?
Yep, that's going to be your antibiotic. So the antibiotic of choice is going to be vancomycin. And I would note that there's a high incidence of multi-drug resistance in this species. Okay, the next species we're going to talk about is going to be C. Pseudo tuberculosis. So it's primarily a vet pathogen, specifically coming from sheep. Causes a granulomatosis lymphadenitis. So it also produces a brown halo on the CTBA, which makes it kind of important because there is a big difference in having pseudotuberculosis than having diphtheria. But the difference is it is urease positive. And so what would C. diphtheriae be?
That was negative.
It was negative, correct. And then on an SBA, you're going to get small, and this one doesn't need to be highlighted. I just haven't taken it off yet. Yellowish, white colonies. But you don't have to sit there for micro and remember every single biochemical reaction for every single species or every single organism. But what you need to know is differential features. So you need to know that like this pseudotuberculosis also produces this halo, but the difference is it's urease positive instead of urease negative. Instead of sitting there and say, okay, pseudotuberculosis has the halo, it's not lipophilic, has beta hemolysis, you know, the urease reactions, the acid production, it produces it from glucose, maltose, but maybe sucrose. Like you don't have to sit there and learn all those for every single organism, just the important ones that can help you differentiate them. Next one's going to be C. ulcerans. And they can also, the isolates can also produce the diphtheria toxin. It's also a common vet pathogen. But you do get occasional outbreaks, and they are due to infections. They're going to be from animal contact. And y'all know what the other one is? Kind of associated with animals too. The other one's going to be your unpasteurized dairy product. And this one also produces a brown halo on CTBA. On your sheep blood agar, is going to have a narrow zone of beta hemolysis. This one's going to be urease positive and nitrite negative. So urease positive, nitrite negative, which is going to be your differential feature from C. diphtheria. So out of all the Cranibacterium, I just took down the ones and went further into the ones that also produce that brown halo, and then how to differentiate them from your diphtheria.
So is pseudotuberculosis going to have a positive nitrate?
Right here, I think. It's variable. for pseudotuberculosis, but it's urease positive. And let's see what's differential between ulcerans. Pseudotuberculosis is variable for nitrate reduction. It's the same. It looks like the difference could be your gelatin hydrolysis, where ulcerans is positive and pseudotuberculosis is negative. But the big part of these is to be able to differentiate it from your diphtheria. I'll stop it after. So the next one's going to be Cranibacterium Ureolyticum. What does that sound like? No. Ureolyticum. If you see that word, what's the first thing that comes to mind?
Urines?
Urine, right? Urea, urine. And so let me tell you real quick, this is their catalase positive. Pause. Urease positive. So urease positive. And nitrite negative. And so urea is ureolyticum is common cause of UTIs. Or you can remember urea. And then it's treated with vancomycin. And you will see pinpoint non-hemolytic white colonies and that characteristic cranioform morphology. And I think that's all the most important things out of your Corynebacterium. So remember your diphtheria is going to be the big kahuna of the Cranibacterium, that they have Chinese letters or club shape. A lot of times it'll sit there and give you a million reactions and then one word about Chinese letters, and that'll be the key to your answer, and a lot of micros that way.